Well, hello everyone. We are live, and thank you guys so much for joining us. This is kind of an impromptu discussion um, with Immutable Destiny. So, welcome, Immutable Destiny. Please feel free to introduce yourself.
Hey, I'm just a guy who goes on streams every now and then. Usually, now that Gur has been streaming lately, I usually hop on there. I just like having open discussions with people, challenging views, and hopefully it's productive dialogue, even if nobody's mind gets changed.
Yep. And my name is David. If this is your first time watching, I'm an undergraduate student at the University of Maryland, Baltimore County. I'm studying biochemistry and molecular biology. So, it is a pleasure to have you with us. And please hit that like button, hit that subscribe button, and hit that bell notification down below. So, formidable. Thank you so much for joining me.
Hey, Ed, glad to be on. Always fun to talk with people and get to see where people are in their journey and what's going on with creationists or whatever people want to talk about.
Yep. So yeah. Remind me. What you're in school, right?
Yeah, so I'm currently in school with a local college to do a certification program in biotechnology. And my bachelor's degree that I earned a few years back is a BS in chemistry.
Okay. Cool. And if you have any questions in the cyber chat, please feel free to ask them. So I guess we can talk a little bit about abiogenesis if you want, or we can talk
Yeah,
about some of
sure.
the stuff. So I guess where we're at with
Abio is always
abiogenesis.
fun.
Yeah, I enjoy studying abiogenesis, even though it's kind of complicated. Um,
That's an
so
understatement.
yeah, very much an understatement. So I guess we could start by some of the creationist straw men about abiogenesis. Um, I guess number one, abiogenesis is not the same as evolution.
Oh, I thought you were gonna go with the whole spontaneous generation thing.
Oh yeah, that spontaneous generation. We could definitely touch on that too. Um,
Yeah, one of the really common objections that I've heard a few creationists bring forward, and it's a really common one, is about how like how is abiogenesis different from spontaneous generation? And that's because like it gets brought up because a biologist a while back, I forget, it wasn't Hegel, I forget who brought it up though. It developed the law of biogenesis, which is that life always comes from pre-existing life. And this idea of abiogenesis, this idea of life emerging from chemicals, doesn't seem to be a really seems to be a violation directly of that. Although the people that developed the law of biogenesis noticed that there was an exception to it, and the exceptions were in terms of diseased cells and in terms of cancerous cells. That cells that have become cancerous due to a myriad of reasons, uh, that there when people are born, there aren't always cancerous cells. Cancerous cells develop um, due to a variety of different factors, and you know it's with abiogenesis. One of the things we have to think about is how life comes about, because according to the fossil record that we have to kind of see where how evolution of life takes place, what we notice is the Earth that formed about 4.2, 4.4 billion years ago. We didn't see life form on the Earth. Like the earliest microfossils that we have formed about what is it? It's like 3.7 to 3.8 billion years if I'm not like horribly far off on that one.
Yeah. So obviously abiogenesis is a pretty big field, and there are many competing models and hypotheses for abiogenesis. Um, so we can just start by touching on some of those. What have we found so far?
What have we found so far? Well, that's a really challenging. There's a lot of interesting research focuses in abiogenesis. One of the ones that came about beyond, I think it was like 1980s, this concept first came about, is this notion of autocatalysis. Like a really big finding that was actually confirmed experimentally by Lee Cronin in 2017, 2018. I'd have to look it up again. But first of all, let me explain what autocatalysis is. Autocatalysis is the idea that if you have a compound under certain conditions, that compound can undergo chemical transformations in such a way that that product A goes to product B, goes to product C, but after some number of intermediates, it connects back to the original thing, right? So the idea is that this reaction eventually feeds back into itself, and it forms this cycle, so to speak. And Lee Cronin was one of the first people who verified this. But the idea of autocatalysis is important because, well, when we talk about abiogenesis, we're not just talking about one compound reacting with one other compound. We're talking about a very complicated, effectively soup of chemical compounds that are, dispersed. Well, there's a myriad of reactions that can take place, but if there's cycles that these reactions can undergo, right, even if the yields are low, the yield is irrelevant because the compounds that the compounds that are present and that partake in this cycle, in these autocatalytic sets and autocatalytic reactions, are going to be the ones that are being continuously regenerated. And so this issue, like one of the issues that James Tour likes to bring up of concentration, isn't irrelevant in that type of idea, because the idea of concentration, this idea of like yield, is one that's related to equilibria. And if something is reacting in a set, well, there isn't really an equilibria, right? Things are always forming and things are always dissipating, right? There's not a very clear way to define, what the yield would be, given that there's a continuous, give and take for an autocatalytic reaction.
Right. So I guess let's start by talking about some of the current models. We'll start with the RNA World Hypothesis. What exactly is the RNA World Hypothesis?
Right. So the RNA World Hypothesis, to my best knowledge, the best way I can state it is that people have the conception that RNA is a very useful molecule, and it is. It really is. It can form structures called ribozymes, so it can perform. It can fold up into these really complicated three-dimensional structures and have reactions that take place. It can undergo self-replication, meaning that if you have a strand of RNA and you have another sequence of RNA that's in a proper sequence in the right geometry, it can cause that first sequence to replicate. So the idea with RNA World is that the first type of life, or the first kind of progenitor stage of life, is one that consists of RNA. It consists of RNA that's replicating in some way. And that somehow or another, right, that later on for the ride comes along things such as the genetic code and things such as metabolism and things like lipids also develop. I'm not entirely sure. I'm not overly familiar with RNA world theory and in terms of how they're trying to rationalize some of these models. Because to me, it seems it's a fascinating idea, but I don't understand. I don't understand how you get certain things like proteins or other stuff to emerge from it, given that if RNA is perfectly capable of self-replicating and doing all these other fun things, why wouldn't the system just continuously use RNA? And why would you have a system that uses this very complex amalgamation of proteins and lipids and amino acids and things of that nature?
Yeah, exactly. It's abiogenesis is pretty complicated stuff. Do you personally have any favorite models that you prefer when it comes to abio?
Yeah, I used to be very strongly sympathetic towards metabolism first. The idea of metabolism first is that in modern cells, we have processes of metabolism that occur. We have things like um, glycolysis and gluconeogenesis. So the process by which sugars in your body are essentially disassembled by molecular machines and transformed into usable energy for the cell by ATP. There's also so fatty acids that occur in the body also undergo another type of process of fatty acid metabolism. And the idea essentially is that the idea with metabolism first relates strongly to the idea of autocatalytic sets. So the idea is metabolism is a process that's always increasing energy in the environment. It's always disorganizing the environment around it. And one of the things that entails, or one of the things that you kind of expect there, right, if you're thinking about abiogenesis in that kind of way, is okay, well, it's not too hard to conceive of having a system that that's just continuously undergoing these autocatalytic reactions and autocatalytic sets. The issue fundamentally, though, is there's only so much material on the Earth, right? So autocatalytic reactions and autocatalytic sets are as useful, are only as good as the amounts of things that are supplying that reaction to go forward. So if it's just occurring in one place, eventually it's going to stop, and that's a problem. The other thing about metabolism first, and the reason I don't hold to it, is we know that proteins are a thing that biology uses. So what are proteins? Proteins are comprised of amino acids, and and essentially what they do is they get formed by a process when DNA is replicated by means of by process of translation in the ribosome using the genetic code. And what happens is, in eukaryotes at least, these proteins become folded, or these chains of amino acids, which we call peptides, become folded, and those proteins then carry on some function for the cell. So it might be to transport a nutrient out of the cell that's not in the cell into the cell. It could be all sorts of things. And the metabolism first models don't usually tend to account for that. So the current model I'm kind of really interested in is a kind of hybrid model between genetics first or what's sometimes called like RNA and genetics type of scenario. And the idea is that you don't have just one thing or another. You don't just have one type of thing occurring. You don't just have metabolism. You don't just have RNA. You have this kind of you have systems that are interacting. You have systems like peptides that can interact with stuff like RNA. And the idea is that, at least with some models that I'm familiar with, like the work of Dimitar Kunev, is that is essentially, without going into too much detail, that this peptide RNA system interacts in a way that is mutually beneficial towards both the RNA strand and towards amino acids and peptides. And so what happens there is you have this feedback system where the peptide protects the RNA, and this RNA can also serve as this kind of pseudo template for allowing amino acids to replicate. And I think that's at least somewhat of a promising start towards understanding the origin of life. There's obvious things that you can ask about it. One thing you could ask is, okay, even if we have this kind of system, right? Even if we have this system where I do have RNA and peptides that exist, what's the system going to do? Is it going to focus on membrane stability? Is it going to focus on some kind of form of primitive metabolism? What is it going to prioritize given this kind of hypothetical? I'm not sure. The model that I'm most familiar with, being Demeter's model, is relatively new in terms of models that have been proposed. It was proposed in, I think, originally 2016. So it's a good question, right? We don't have all the details fully fleshed out. So we'll wait and see. We'll see how some of these hypotheses how some of these hypotheses develop and carry on.
Yeah, exactly. Though we are, of course, pretty far from understanding the origins of life. Dr. Mark Pollack, good to see you, my friend. Not able to stay at one for long, but we'll listen to it when you post the whole thing later. RNA World Origins of Life are fascinating. Yeah, I definitely agree with you, Dr. Mark. It is very quite fascinating indeed.
Yeah, I think they're fascinating. My biggest concern with them is, like I said earlier, it's fine that you have RNA that's replicating and that it has this breadth of chemical diversity and chemical reactions that it can perform. But again, if you have all this, right? If you have all this complexity and all these great things happening, what is it about the system that's necessitating or that's favoring the system to incorporate things like lipids and amino acids and proteins, right? Because that's something we know that life has. And that's the really odd, to me, the kind of seemingly paradoxical aspect of those hypotheses is that they're very interesting, but they don't seem to really it's not very clear how they're going to explain proteins and stuff like that, given
Right.
what they're starting with.
Another thing is that RNA is a pretty unstable molecule.
Yeah, RNA, to my knowledge at least, it's susceptible to hydrolysis in water. So that's led some people to believe that I think the work of Damer and Deemer have kind of published work not directly related to this, but related to this problem about how are you going to have RNA? How are you going to have like these polymers? What is a polymer? A polymer essentially is just it's a sequence of something. So it can be a sequence of amino acids, it can be a sequence of nucleotides, and it's longer than one in length. Sometimes it's 10, sometimes it's 15, sometimes it's 8. It can really vary. But with what we were asking, right, like with what you pointed out, RNA is susceptible towards hydrolysis in solution. Well, okay, if that's the case, then how are we going to do this? And one of the solutions that's presented to it is, well, let's not just consider a aqueous phase solution, right? Let's consider something that's fluctuating between environments. That's fluctuating between something like between wet, dry environments.
Yeah.
Right.
Dr. Pollock, I guess my answer would be changing world around them. RNA alone can't stand up to the environment well, and developing novel tools with greater stability, e.g., amino acids, lipids, and DNA, can address some of those difficulties while losing some of the capacities.
I guess that could work. It seems, I don't know. It in some sense it seems a little post hoc, right? Because it seems that you're using other things to explain why RNA is the thing that's being preserved and more stable when in fact, right, RNA does do a lot, right? It does in terms of what we know about its function for cells. But really, it's peptides and proteins that do quite a lot of the action in terms of chemical responsibility.
Yeah.
Proteins are the thing that's responsible for catalyzing, the reaction of aminoacyl tRNA synthetases, right? That reaction recognizes a particular strand of RNA in order to get the amino acid close enough in range to undergo a reaction to make peptides. But largely speaking, it seems that proteins really have a lot of the responsibility in terms of chemistry. To me, I don't know. That seems to indicate to me that there's something else happening here, right? That it could be that RNA is the most important thing. It could be that RNA is really the taking all the responsibility in the beginning. But if that's the case, again, wouldn't you expect it to be the case that the types of reactions that occur in cells nowadays are mostly performed by ribozymes and these very complicated sequences of RNA? And that's not what we see. We see proteins, by and large, catalyzing chemical reactions.
Yep. So, how far do you think we are from understanding the origins of life?
Oh, I'd say we're a long way away from it. I would say, geez, it's hard to even put a number to it. Maybe we'd be close to it in another like 50, 60 years from now. It's hard to say because
Yeah.
there's a lot of conceptual barriers. So the first barrier is kind of getting everything in the right place, right? Is kind of getting things in this order in this proper kind of configuration so things can start undergoing an evolutionary process. And it could be, by the way, to correct myself a little bit, there are some people that think that these model systems, that these what are sometimes called protocell systems, these systems that aren't quite cells but exhibit some properties that cells do have, could undergo some form of evolution, in the sense that, for instance, if I have sequences of amino acids that are really stable and that are also stabilize, say, the cell membrane of a cell, right, or a protocell in this case,
Right.
and that process survives replication by some means, say it's replicating due to hot and cold cycles near a hydrothermal vent or something like that, and that protein survives, or that peptide survives, right? Maybe there's a form of selection that's occurring there. Maybe there's some kind of very very different form of selection that's able to take place. That's possible, but I don't think it's really a viable model unless you're taking into account something really critical, which is, how do cells nowadays ensure that they maintain information, right? it's not a random process that they use. The process that they use is via the genetic code. It's via this complicated process of replication and transcribing codons into transcribing a sequence of codons into an equivalent sequence of amino acids, and having those amino acids perform some chemical activity. So to me, I think really any model of abiogenesis, or any way that we're going to talk about this, we have to have some kind of form of genetics, or we have to have some kind of system at the very least that's in a way remembering, that's able to remember information or store information, right? It's probably almost certainly not going to be as complicated as the genetic code that we have currently, but maybe there's some primitive version of it, or maybe there's some weird analog system in prebiotic conditions that does good enough until such a thing can evolve later on, when cells have enough energy and enough resources to play around in a proper evolutionary sense to develop that type of thing.
Yep. So yeah, a lot of pretty interesting stuff.
I think there's another thing that's really critical too, which is, I'm making replication sound like it's this really important thing, but keep in mind, right? And this is a point that creationists will, will bring up. Oh, well, it has to be, like perfect replication. No, it doesn't, right? A system it needs to be able to replicate. And whether or not that replication is perfect or not is irrelevant to the point of abiogenesis. What's going to survive are the things that are stable. And the things that develop, the things that are more stable when they replicate, or the things that are more stable as this process continues, are the things that are going to give rise to some form of life. In this case, the form of life that we have that uses proteins for their chemistry and DNA as its genetic material.
Yep. Yeah, I see your point. I wonder if that's indicative of where early life started, or just where stable life landed. Hard to say, and we may never know for certain. Yeah, there's a lot of things we will never know for certain, and I'm of the opinion that we'll never know exactly how life started. Um, it's always amazed me just how much information can be stored in 5 bases of nucleotide acids, 4 in RNA, DNA, respectively. Wouldn't be at all surprised if early life functioned with it too. Yeah, I would agree.
That's a really fascinating point, and what's more fascinating about it, and this is a point I've brought to creationists because I learned about it only a few months back by talking to abio researchers, is take a look at the genetic code. Actually, what do you notice? And there's something interesting to note about the assignment of amino acids. The assignment of amino acids and the types of codons that are used. What do I mean? Well, it turns out to be the case that some of the amino acids that are used, in fact, some of the more basic amino acids that are used, not basic in terms of chemical reactivity, are actually, let me see if I can pull up a codon table to make the point. Um, the point I'm making, I'm sorry, my brain is really is a little tired, but the point I'm making is this: is that some of the amino acids that are used, right, especially some of the ones that are kind of responsible for protein structure and certain motifs that we see, are in fact GC rich.
Yeah.
Let's see if I can share it. Well, yeah, let's see if I can share my screen. Maybe I can. Let's see, share. see. Let's see, can I find... let's see, can I find a decent one? Actually, no, you know what, it's okay. If you guys want to, you can look up a codon table. There's one that I like to use, but I can't find it at the moment. But yeah, what's interesting about codon assignment is the fact that a lot of codons that are GC rich. So if you go hunting in the codon table for codons that are GC rich, a lot of them turn out to be ones that specify amino acids like such as alanine, glycine, proline, aspartic acid, and arginine. Aspartic acid and I think glutamic acid are the ones that are GC rich. Keep in mind, there's 64 codons in the genetic code, so if I'm wrong on that, my apologies. Feel free to call me out on the comments there. But yeah, it's an interesting observation that some of the GC rich some of the rich GC. I swear I can't speak today. Gosh, what is what is wrong with me? That some of the GC rich codons are ones that are responsible for quite a bit of amino acids, and in fact, GC rich codons specify amino acids that are abundant or that are redundant, rather.
Right. So, anything else you want to talk about?
Do you have a favorite model for abiogenesis, or kind of any thoughts as to
I guess if I
the
had to pick,
field?
I guess if I had to pick a favorite model, it would probably be the Rna World Hypothesis. But yeah, there's definitely so much that we have to learn about the origins of life. I think that it is very much worthwhile to do origins of life research, and it is certainly going to help us understand our place in the universe a little bit more. I don't think we're anywhere close to understanding the origins of life, and I don't think we ever will. But the best we can hope for is various models and testing which models are viable and which ones aren't. And
Yeah, I think
that's the interesting.
I think the origin of life studies also do something really interesting as well, which is while it's not possible, while it's not possible for us to understand perfectly how life came about on Earth, because it's already here, right? What we do know, given astronomical data, is that there are some planets in the solar system, some of the moons of Jupiter, for instance, Europa and Titan, I believe, are two moons that are very promising in terms of their potential to store life, mostly because they have very interesting aqueous environments. Europa, I believe, actually has an ocean of liquid methane, which is actually rather interesting. It's a really liquid methane exists in a very cold state. But the idea is that abiogenesis itself, right? This idea of exploring chemistry that can give rise to systems that are lifelike, well, that's important because we do need to understand and we do need to have an idea of chemical signatures on not only on Earth, but also as a general characteristic to help us see if there's life anywhere else on the universe. Because even if we can't see it, it would be interesting to see if it's out there, right? And how would we know if it's out there, right? There's open questions regarding, uh, you know, if there is if there is life out there, does it use similar genetics systems than we do? Does it use similar proteins to the ones that we use? Does it use the same nucleobases and nucleotides that we use? These are all really fantastic and interesting questions.
Yeah, exactly.
I don't
And
know. I don't know if there's a really clear-cut answer, because obviously Earth at least, we have a sample size of 1, and it's a very likely. It's a very you know it's quite possible that life is a very rare phenomenon that only occurs under select conditions. But I don't know. I don't think. I think that's a little naive, and probably life exists elsewhere in the universe.
yeah, I think with billions and billions of galaxies, each with billions and billions of stars, and each with dozens of planets, the probability that life exists elsewhere in the universe is probably very high. Though I guess this also begs another question: what exactly does it mean for something to be alive? Are viruses alive, for example? I know Dr. Pollock in the chat says yes, they would be. He does consider viruses to be alive. Others, of course, would disagree. And I would say that I would have to agree with Dr. Mark there. Um, I would define life as any self-replicating system that has the ability to undergo evolution and is subject to natural selection. So that's how I would define life.
Yeah, there's actually a really interesting paper I remember reading about. Yeah, here it is. Maybe if you want to put this into the description or something, this would be a fun little paper to read for people. But here, let me show it. So a few years back, and the reason I found this paper was I listened to a podcast that Sean Carroll does, and the name of his podcast is called Mindscape. And about a year ago, I think, maybe it's a little bit further back than a year, he interviewed a guy named Stuart Bartlett, and he wrote this paper defining life in the universe from 3 privileged functions to 4 pillars. And this paper is a little bit lengthy. It's 22 pages, so it's there's definitely a lot to unpack with it. I read the paper some time ago, and I would like to read it again. But the idea here is that it goes into a few things. So it goes into kind of the stuff we were talking about in regards to replication, metabolism, and kind of this idea of compartmentalization that's occurring in life. It compares it's talking about bottom-up and top-down approaches that we use. But it also, one of the main purposes of this paper, it tries to give an idea, a general kind of, it attempts to give a general definition of what life is. And it does that using these four basic, these four principles. Needless to say, this paper it's mildly technical in the sense that there's definitely a lot that's going on with it. It's bringing up concepts that are specific to the field of abiogenesis, but also fairly well understood in genetics and biology. And it's interesting, at the very least. Even if you're not convinced by the paper, I would give it a read, because this type of paper, even though it's conceptual, is one that's very important. Is one that's very important to understand, because one of the things it's trying to do is one of the things we should try to do to understand abiogenesis, if we're wanting to understand the origin of life, right? Well, what is life, right? what is life as a general characteristic of the universe? What does that mean as a concept? What does that mean as a property of the universe? We know things that that electrons have electric fields, and if I move an electron in some direction, that change in electric field causes a magnetic field and things like that. But we don't have as compact a definition for life in the way that we do with some of those other things. So, there is room for improvement, and there is a very serious discussion to be had about conversations focused around, let's not worry about, let's not get too wrapped up in the science. Let's also consider a general idea of what life could be considered and how to identify it in the universe.
Yep. Dr. Pollock, GC rich portions of DNA do get interesting. The richer DNA RNA in these bases, the higher the melting temperatures of the said DNA RNA. There are also let's see here. There are also regions of DNA that are rich in these called CPG islands, have lots of interesting functionality to them. The borders between viruses and life gets even harder to define with some things like the giant viruses I was talking to Dr. Dan about. Yeah, I definitely agree 100%.
Yeah, that's actually one of the so when you talk about like hydrothermal vent theories for origin of life, that's one of the reasons that people suggest that that CG rich sequences were probably the first sequences that life used, but not too much of it. And the reason is, imagine like, for those of you that remember reading the story of Goldilocks and the Three Bears, it's a very similar type of story, right? If you have an abundance, if you have too much GC rich sequences in whatever kind of nucleotide strand you're interested in, well, what's going to happen to it? Well, it's going to be too stable, and it's never going to be able to replicate itself, right? These base pairs that allow for this helix to form, it's just going to stay there, and it's not going to allow anything to interact with it or allow it to replicate in some meaningful way. In the same sense, in a similar way, if you don't have very many GC rich sequences, well, what's going to happen? Well, if there's not a lot of GC rich sequences, there's not very much chemical energy trapped or stored in that sequence of nucleotides. And, well, that means the sequence is going to be really susceptible to degradation via heat and all sorts of other stuff. But if it's just right, if there's a decent mixture of GC rich nucleotides present in whatever nucleotide strand you're considering, well, it'll be able to replicate at certain temperatures. But once that temperature range is, once that you're lower than that temperature range, the nucleotide sequence will fold back in on itself, because there's not enough energy anymore to cause that sequence to be vulnerable or susceptible to chemical degradation.
Yeah. Hey, Dr. Dan, melting temperature, we just talked about that. Melting temperature equals the temperature to break apart strands, which matters a lot. So yeah, it's very interesting stuff.
It really is, and that's the thing, that there's a lot of perspectives to understand, to try and understand this theory from, or to understand this hypothesis from. There's benefits to having top-down approaches, right? There's benefits to looking back at, to looking at what we have currently and trying to, scrape away what's the messy bits of it to try and find the most likely candidate for simple cells that could have been present in in whatever prebiotically plausible environment you want to envision. But similarly, right, having top-down approaches is also a little bit more is a bit more realistic and also more challenging for the reason that in a top-down in a bottom-up approach, we don't know all the chemistry that's accessible to the system, right? And we have to test things one by one. We have to be, very meticulous about the kinds of systems that we're developing, and a lot of the ideas that we're going to have in regards to what chemistry is plausible or not is going to have to be based upon what we understand from the geological column, right? The very early history of the Earth during the Hadean period is going to inform us on what kinds of chemistry were and were not plausible.
Right, exactly. Dr. Dan says, going to bed momentarily, but man, this sounds fun. People start so late. Some people have kids and are tired all the time and need to go to bed.
Well, Dr.
Yeah.
Dan, I mean, if you want to, I would love to talk to you about this as well, because you are far
Oh,
more
yeah.
educated than I am on this, and
Yeah,
it's
and
always
Dr. Dan,
interesting having spats about abiogenesis and what kinds of things we can learn or potentially theorize about it.
yeah, and Dr. Dan, feel free to come on and talk, and we can just talk abiogenesis for a good hour or so. That'll be a lot of fun, also.
Yeah, I really do want to. I do want to read like some of the more diverse papers out there, because there's some people out there that really are very creative in how they're approaching this topic, and like I said earlier, I have tremendous respect for for their efforts and how ambitious they are trying to solve this issue, because between this and trying to understand what was occurring prior to the Big Bang, these are one of the two biggest gaps that we have left in our scientific understanding of the world around us.
Yep. And, yep. And I definitely agree with you there. Anyways, I am getting tired and I need to get
Oh
to
yeah,
bed.
man. I got work
So,
I got work like 7 or something like that, so I
yeah.
probably should get to sleep.
Yeah, same for us to get to bed. But anyway, yeah, thanks for having this impromptu conversation with me.
Hey, no problem. Glad to be here, man. Always fun to
Yeah.
to talk and not be worried about having people hold a flame to to what I'm saying being politically, or not politically, but trying to always make sure I'm correct.
Yep. But anyway, see you all later. Thank you so much for tuning in. Don't forget to hit the like, subscribe button, and the bell notification down below. And if you have not yet gotten your COVID vaccine, get vaccinated. So,
Amen to that.
yep. And if you qualify for your booster shot, don't forget to get your booster shot.
Yep. Stay safe. Don't spread this virus more than it has been.
Yep. We are out. Good night.